everyone, welcome to my third video in this series about mutations and disease, and today we're going to be looking at some key terms. So the first key term that we're going to look at is a gene. And so a gene is a section of DNA that codes for a particular protein. And so within a human you have roughly 23,000 genes. And so obviously each one of those genes codes for a specific protein or making part of a protein or a polypeptide chain. And so you have here an example of this, and so here you've got your basic structure of DNA, and these are your nucleotide bases, these bits in red. And so on an average gene, so for instance if we were looking at the CFTR gene that codes for the CFTR protein, that's made of several thousand of nucleotide bases. So a version of a gene is what's known as an allele. And so for instance, if we were looking at cystic fibrosis again, you might have the allele for being healthy, and you might also have the allele for having cystic fibrosis. And so you can get many versions of genes. And so for instance, eye colour is a good example of this. You can have many different versions of a gene for different eye colours. And so you can see here the position of these alleles on the chromosomes, and you can see these here in the same position, which you'd normally expect to find on a chromosome pair. The next term is the genotype, and that's the combination of alleles an organism has. So for instance, they're usually represented by letters, so big B small b or big F small f. The phenotype is how that's expressed, and so how that's expressed within the organism. And so blue eyes, for instance, brown eyes, whether you've got cystic fibrosis or not, whether you don't have cystic fibrosis, are all examples of phenotypes. A dominant allele is one known, or if you have a dominant section, is only when one allele needs to be present for the characteristic to be expressed. And so in this case it's always represented by letters that are uppercase. An example of this might be if you had the dominant allele for eye colour, which was, let's say, brown for example, and then the recessive eye colour, which might be blue, you'd only need one brown because it's the dominant one for it to be expressed. Which takes us on to recessive, when both alleles need to be present for the characteristic to be expressed, and so you need two of them. So an example of that is cystic fibrosis. You need both cystic fibrosis alleles in order to have the condition. A homozygote, that's where you have, for a particular characteristic, you have the two alleles which are the same. So for instance, big C, that could also be small c, or big F. Some examples of that. A heterozygote, that's where the two alleles for a particular characteristic are different, so big C, or big S, small f. And now we come to a monohybrid inheritance. That's a genetic cross where you only consider a single gene at a time. And so for this qualification, we only ever look at monohybrid inheritance. And so an example of this is a monohybrid inheritance for two carriers of cystic fibrosis. Now, the previous video we went over some of the symptoms and how cystic fibrosis is caused, and so it's an inherited disease, and so it's received from your parents. And so here we're going to go through what a genetic cross would look like for two carriers of cystic fibrosis. So what this leads on to is the idea of the alleles that cause cystic fibrosis. Here, the large one, F, that's healthy, so that's the dominant one. The small f is the one that causes cystic fibrosis and is recessive, and so in order to have the condition you need both recessive alleles. And so you can have a carrier for this disease because you can have the genotype big F small f and be a carrier for the condition and not have any of the symptoms of it. And so the first line that you always write down will be what the alleles are, and then the F1 phenotype for the people being crossed, and so in this case they're both carriers. You could also possibly put these as healthy. The F1 genotype, that would be big F small f in both instances because they're carriers. And now the next line down is the gametes, and so this is what the sex cells contain. And so in all sex cells they only have half the genetic information, and so in half the gametes, for let's say the father, half the sperm will contain the healthy allele and half the sperm will contain the ones that contain cystic fibrosis. And the same is true for the mother's eggs. So in this instance, half contains the healthy allele, half for the recessive cystic fibrosis causing allele. Here you have a Punnett square, I've just drawn the gametes along the top two here and the gametes for the father's sperm here, and these are the possibilities that you can get for this genetic cross. These are the different genotypes that you can get, and so that gives us an F2 genotype, and that refers to the second generation, the F1 being the first generation. You can get these possibilities, so you can get an individual with a genotype big F, big F. You can get two individuals with big S, small f. Just flick back to that. The reason why it's two is because, well, there's two of them there. And then you get one individual that has the condition. And so the F2 phenotypes in this instance will be one healthy, corresponding to this one here, two carriers, these two here, and one sufferer, these two here. This would give you a 25% chance of producing somebody who's healthy, 50% chance you'd expect carriers, and 25% chance you'd expect sufferers. Now this is always done as by chance. It is possible for two individuals who are carriers for this disease, in order to have, they could have 4 or 5 kids in a row with cystic fibrosis. That would be unlikely because there's a 1 in 4 chance of those two individuals producing one child who has cystic fibrosis. But these numbers give an overall estimation or a probability of the likelihood of that outcome.